virology block, but if you look at the slides, you'll notice we're covering bacteria today. These are sort of just the random ones, the atypical bacteria we didn't cover in block 3. So usually takes about 2 days to cover this deck, and then I'll post the virology deck, which is the bulk of block 4. It's a big deck, it's like 180 slides, 170, 180 slides. Yeah. So.
Bring it.
Yeah, that's great, bring it.
You did it once.
Yeah. Oh, that's great. We already covered a lot of ground. One of you guys was telling me you're counting the number of organisms that we cover, which is super cool. Like, I think that it was like 65 depending on how you slice all the stereotypes and generalities, the different taxonomic levels we covered. But these are some of the major genera that we've covered in block 3. We've just hit a tremendous number of organisms already. It's certainly not comprehensive. You'll hear about other things in other classes as well. But with the atypical bacteria we need to cover, we've got some really significant pathogens, chlamydias, mycoplasmas, coxiellas, rickettsial diseases that we'll cover. So these we'll take care of today, and then tomorrow we will start talking about acid-fast bacteria. So these also, the commonality is that they, both atypical bacteria and the acid-fast bacteria, do not stain with gram stain. This latter group includes Mycobacterium tuberculosis, which is a major pathogen. We'll spend the bulk of Wednesday talking about TB, and then most of this block is virology, and the last day is mycology. So it kind of gets a little bit underserved, in my opinion, but just kind of like wave tops for mycology and fungal diseases. Here's our little organizational schema of what we're covering. So atypical bacteria, chlamydia, coxiellas, rickettsial species, and then the mycoplasmas are weird in that they have no cell wall at all. We'll talk a little bit about their membrane. Yeah, very weird. But they're still, these are all still considered bacteria. They all still have a 70S ribosome, 50S, 30S. Common features are they do stain poorly. A lot of these guys are intracellular. Many of them have reduced genomes because they rely on host cells for energy or from other components. So they do have some common features, and that's why I like to group them together. Okay, so we'll start off talking about this family for which chlamydia belongs, the very famous Chlamydia trachomatis, STI, but also includes some other members that are clinically important as well, such as Chlamydia pneumoniae, different species. This family is called the Chlamydiaceae. Like all these other atypicals, or most of them, these are obligate intracellular parasites. So a couple of important implications to connect to that. One is when you are treating them and you're using an antibiotic to clear up an infection caused by one of these intracellular parasites. Unlike extracellular bacteria where the antibiotics basically just have to go into the bacteria that are outside those cells, if you treat an intracellular pathogen, it has to go into the cell also. So there's another barrier to get through. It makes it a little bit harder for treatment purposes. Another implication is for the immune system itself, and we're talking about intracellular parasites and antibodies being able to access them and clear them. If you're an intracellular parasite, that doesn't work so well. So that whole cell-mediated arm of your immune system is relied upon more heavily, and the humoral side, which involves antibodies, is less important for a lot of these intracellular bacteria. Cell-mediated immunity, super important. Also, because of the fact that they rely on the host cell for various things, as I've mentioned, like ATP perhaps, or some other biosynthetic pathways, because of that reason, it is harder to grow them up like conventional media. Like chlamydia, you would have to use a cell line, like a eukaryotic cell line, in order to grow them. So for that reason, diagnostics is harder with these guys, and we are increasingly relying on PCR in order to identify them. They are, in terms of the shape of the Chlamydiaceae, these guys tend to be pretty round shaped, very, very small. They would be considered gram-negative. We'll talk about that more in the next slide. But with this small size, historically they were considered to be viruses initially. Because of that small size, you could filter them, and that was a characteristic of viruses. But we we know them now to definitely be bacteria, but it's still very, very tiny. You may not be able to see this in this stain, but we're looking at some epithelial cells from the erythrocyte, and the bacteria are in those cells. This is not a gram stain, this is called Gibson stain. So very, very small and hard to see, even with those specialized stains like Gibson and Pigmented stains. With these two pathogens that we're talking about, Comotus and Pneumoniae, both of these pathogens are strict human pathogens. There is a zoonotic one called Sataki, which is really famous because it infects parrots, and it's a sexually transmitted disease among parrots and other birds. And it can actually, the only reason why I say that is because you can actually get people sick as well. Strange, but we're not going to talk about psittacosis, the disease caused by Chlamydia Sataki. Yeah. Yeah. Yeah, I guess that's common knowledge. Yeah, I have to admit I have not brushed up on that recently, but there is an association with that.
How do the humans get it from the parents? Well.
John, you have an answer for that? Anyone else who's like, oh.
When a person loves a person.
Someone want to tackle that one? No, I honestly don't know. It's
We haven't done reproductive yet, so.
But I can look that up. But fortunately, we're only talking about Comotus and Pneumoniae. These guys. And these guys are both relatively easy because their reservoir is strictly the human population. You're not going to get them from your pet koala or your pet parrot. Also, because they're maintained in the human population, that means a lot of these infections are going to be subclinical. Not all of them, you can definitely still get sick, but generally speaking, some of the more virulent pathogens that we talk about have reservoirs outside the human population, because if they were maintained in the human population and they were super virulent, they would burn out pretty quickly, as in they would kill us and they wouldn't be able to sustain themselves. But these guys do very nicely in the human population because oftentimes they're subclinical and they are associated with very low mortality rates. Okay, in terms of the cell structure of Chlamydiaceae, they are considered to be gram-negatives, but they've got some distinctive features. One is, so they have the inner layer and the outer layer of the cell wall that we've talked about, so that phospholipid bilayer. But in the middle, we're used to seeing a peptidoglycan layer for gram-negatives. These have a peptidoglycan-like layer in between, but it's not exactly peptidoglycan. And the only major implication you need to know about that is it just makes it very, very difficult. If you were to try and stain one of these tiny bacteria anyway, you might get like a opaque color, or they might not stain at all. So again, for that reason, we use other stains if we want to stain them. Also, the LPS has very weak endotoxin activity associated with it, unlike some of the other gram-negatives we've talked about, like E. coli and Salmonella, have much, much stronger endotoxin activity. And then importantly, in their cell wall, they have these MOMPs, major outer membrane proteins, that help compensate for some of the structural weaknesses in that cell wall. And they're also important for epidemiological purposes too. When we start talking about Chlamydia trachomatis, and we talk about different serovars of Chlamydia trachomatis, this is the reason why they're different serovars, is this MOMP protein. So it's important for a couple different reasons. Lack of rigid cell wall, that sort of ties in some of these other points. In terms of eukaryotic features, eukaryotic-like features, it does have cholesterol in the cell membrane, and it gets that cholesterol from us, from our own cells, so it doesn't have synthetic pathways to make its own cholesterol. It'll steal that from us. The implications there is it changes the rigidity and the structural integrity of these cells. In terms of prokaryotic-like features, they do still make their own proteins, but again, they have that 70S ribosome. They are sensitive to antibiotics that would target protein synthesis. So, you guys remember any of those antibiotics that we talked about that target the 50S subunit or the 30S subunit? Yeah, which ones target the 50S, the big one? Macrolides and glucosamide, right? and then the small one is tetracyclines and aminoglycosides, right? So those work just fine on these guys. Fluoroquinolones also work well on these, just FYI. What do fluoroquinolones target? DNA gyrase. Yes. Okay. So they are susceptible to a bunch of different antibiotics, but we'll see this with others as well. They do have some, because of their cell wall, they might not be as susceptible to cell wall targeting antibiotics. They have a very unique life cycle that's worth studying because it gives you a little bit of hint about how they're transmitted and sustained in human populations. So when you transmit, let's say you're talking about say we're talking about chlamydia trachomatis with the STI, what causes chlamydia urogenital disease. When these bacteria get into a susceptible host, they are in a unique form called the elementary bodies. It's an infectious form. So this cartoon or diagram on the right-hand side sort of shows you how these things work. But this infectious form is small, relatively small. It's like 300 to 400 nanometers, pretty tiny. And it is relatively resistant to the environment, almost spore-like. That allows it to be transmitted and survive for a little bit of time outside of the human body. Also, it is able to attach to, as adhesins, attach to target cells, epithelial cells, and trigger them to engulf those bacteria because they are intracellular. So they get engulfed into those epithelial cells in the urogenital tract, if we're talking about trachomatis. And then shortly after they become engulfed, they start expressing a few genes that convert them into this replicating body called a reticulate body. So the RBs are the replicative form. They are metabolically active. Much larger. They're like twice the size, 800 to 1,000 nanometers. And basically what they do is start dividing in these inclusion bodies that form into cells very quickly. So that's their whole purpose, is to do whatever limited metabolic activities they have and replicate and divide. This electron micrograph on the bottom left shows you one of those inclusion bodies in a cell. So you can see some other cell features. There's a nucleus, there's mitochondria, and we see a mixed population of EBs and RBs. As those RBs start to get dense enough, the health of the cell will start to fail. So this is cytotoxic to the cells, and that triggers those reticulate bodies to start becoming elementary bodies. So that you're just about ready to lose your home, the cell is dying, and you need to become that small elementary body. So they turn on those genes, become small, metabolically inert, and infectious, and then that whole cell will lyse somewhere on the order, usually between 3 days after infection, 2 to 3 days. So it takes some time for this to happen, but when that happens, you get a burst of infectious little elementary particles or elementary bodies that are released. Unusual for bacteria to do that, and kind of virus-like too. So reminiscent of viruses. Okay, so that's common to all the members of this family of Chlamydiaceae. Now we're talking specifically about Chlamydia trachomatis. In terms of the name trachomatis, you guys ever heard of the root trachoma before? You know what trachoma refers to? Anybody?
The mass?
No. It has to do with an eye infection. It was named for this eye infection, and yet it's more often associated with the STI because it's a really important STI, chlamydia trachomatis. But it's very, very important as a cause of trachoma, which we'll talk about in a slide or two. It depends which one of these serovars it is, and those serovars depend on which of those MOPs, those major outer membrane proteins, are expressed. So if it's one of those A, B, C serovars, those are associated with this eye infection caused by chlamydia trachomatis. Serovars E through K are associated with the classic STI that we're mostly all familiar with when we think about chlamydia. And then finally, serovars L1, L2, and L3 are associated with this really aggressive, invasive, kind of chronic STI called lymphogranuloma venereum, or LGV. Each of these serovars have a distinctive tropism. Do you know what the word tropism means? limits the propensity to infect a specific cell type or tissue. So if you have tropism for unciliated urogenital epithelial cells, for example, then you probably wouldn't be able to infect a ciliated respiratory epithelial cell. So they have specific cell ranges that they can infect. Mucosal membranes of the urethra, endocervix, endometrium, fallopian tubes, respiratory tract, and conjunctiva on the eyes, that really thin layer that protects our eyelids and the white surface of our eyes. In terms of immunity and pathogenesis, unfortunately, we do not get long-lasting immunity when we're infected with chlamydia and trachomatis for a number of different reasons. It doesn't usually elicit a very strong infection, especially during your first bout with one of these pathogens. Also, unfortunately, if you get reinfected, you will have enough of an immune response just to get strong immunopathology. So it's kind of like the worst of both worlds. You don't get protection from future disease, and you also have a more severe pathology if you've encountered that pathogen before. Very unfortunate. But that immunopathology, the body's inflammation response to these pathogens, is what's responsible for a lot of the pathology with all of these different cerevires. So we'll talk about the eye one first, trachoma. This is a big problem, not so much in the United States, but certainly elsewhere in the world. Leading cause of blindness, at least in terms of infectious etiologies of blindness. It won't compete with cataracts as an etiology of blindness, but probably the number one cause of blindness worldwide from an infectious etiology. And how it does that is basically you get an infection, you have a transmission of those discharge from your eye when it has those bacteria in it, gets transmitted to another person, and you get this inflammatory process that causes scarring on the conjunctiva. And over time, some of that scarring can lead to these fibrotic regions in your eyelids forming, which irritates the corneal surface, and it also causes your eyelids to turn inwards, which, as you can imagine, will also scrape the hell out of your cornea. All of that over time, if it's not treated, either surgically or with antibiotics or however, will lead to enough scarring where you can eventually go blind with this. So you can see here that I used to have a lot more pictures in this deck, but I took them out for slide economy. You can see the conjunctiva is getting really reddened and inflamed, scarring a little bit, and then over time you can see the eyelids turning in on this patient at the bottom there, and how much irritation that would cause to the cornea. So, mention that this is transmitted by just contact, like if you're touching your eyes and you're not thinking about what might be on your hands if you haven't washed them, but flies are also big vectors for transmitting this mechanical carriage and transmission from flies. Towels, clothing. WHO has this acronym that's good to know, it's called SAFE. Talks about just various facets of dealing with this disease, but surgery for this trichiasis, that's the turning in of the eyelids, antibiotics if needed, facial cleanliness, keeping your face clean, environmental improvements. So yeah, those are the basic elements of SAFE.
Someone would have to cry.
Second?
Do we have to cry for the eye insurance?
No. So if you have a bit of a weeping of your eyes, right? Not necessarily crying, but just like your eyes are tearing, there would be bacteria shed into those tears. And if you're not washing your hands, you can have some limited foam-like transmission perhaps. Somebody else's, or if you're touching someone else's hands and they haven't washed it, and then you go and touch your hands, you can transmit it that way as well. So yeah. There's also, kind of on that vein, when we're talking about these same cerevires, A, B, A, and C, there's an important disease that can happen in neonates called neonatal inclusion conjunctivitis. And this is when those babies, kind of like we were talking about with gonorrhea, right? We're talking about babies born to mothers with active gonorrhea, how they can get a really nasty eye infection as they're being, during delivery. Same route of transmission here. The mother would have an active case of chlamydia, the urogenital form, and as the baby is being born, their eyes are susceptible to this infection. So when the infants get it, this is a good picture of, like, you can see how much discharge is coming out of this infant's eyes, so this would be infectious as well. 5 to 14 days after birth, usually pretty good rate of transmission in infants who are born to mothers who have chlamydia infections. We do try to screen for chlamydia. Mothers are pregnant, so we can treat before the baby is born, but if the baby is born and they develop symptoms of this neonatal conjunctivitis, you can treat that with antibiotics. I'm not sure like which one specifically, but you can treat that without permanent damage to the eye. Similar to other diseases that can be transmitted to newborns, if they aspirate this bacteria, if their mothers are carrying it, they can also get pneumonia as well. This is a pretty high transmission rate, 10 to 20%, so that is concerning as well. The pneumonia that's caused by, that affects neonates is distinctive. It's sometimes described as staccato, or machine gun fire rapidity of coughing, very distinctive cough pattern that occurs with chlamydia pneumoniae, or I'm sorry, chlamydia trachomatis with these infants. Okay, on to the serovars of chlamydia trachomatis that can actually cause the urogenital tract disease. So we're talking about which serovars again? D through K. Yeah, serovars D through K. This is probably the number one most reported STI in the US. It will not rival something like HPV, human papillomavirus, genital warts, because a lot of those are less commonly reported, but most commonly reported, above gonorrhea even. We have, this is probably on the high side, but 3 million cases a year in the United States, maybe as low as 1.5 million. 50 million cases globally. We have a lot of underreporting because unfortunately, what did we say about chlamydia infections writ large in the human population? Many of them are subclinical. So people who are infected have absolutely no idea that they're infected. So because of that reason, this is probably significantly underreported. And its concurrency with gonorrhea is just usually because there's no reason why you can't get both diseases transmitted at the same time. So if somebody already has gonorrhea, they can just as easily pick up chlamydia as well. In terms of the pathology, urethritis in males, cervicitis in females, you can kind of see this is a cervix, normal cervix, and an infected cervix that has that really thick yellowish discharge. You can see speculum exam, some of that discharge. Probably good to treat these. They don't really resolve on their own, so good to treat, but the more important reason for treating them is to avoid complications that might develop later on. Complications like ectopic pregnancy, abortion, premature birth, infertility, or PID, which can lead to infertility. Pelvic inflammatory disease, PID. Now, last but not least, we have serovars contribute to this really severe invasive form that I mentioned, the LGB, lymphogranular venereum. Serovars L1, L2, L3. We don't see this presentation a lot in the United States. It's associated with higher prevalence in places like North Africa, Southeast Asia, some places in Latin America. But travel history or high-risk sexual behavior might tip you off. In terms of the presentation, there is a primary stage where you might just have a small papule that would go likely unnoticed, and it's transient. What does that mean? It's a transient papule. It's not going to stay there forever. It's going to go away. So as it goes away, usually 2 to 5 weeks later, patients can go into the secondary stage where this is no longer something you can ignore. Like you might get away with not noticing symptoms in the primary stage. But in the secondary stage, now we have involvement of the lymph nodes, of the inguinal lymph nodes. And we have much bigger manifestations here. On this particular patient, what you can see visibly is there's some edema going on, and there's some swelling in those inguinal lymph nodes. They become very hard, and those are also referred to as buboes. What else have we talked about in this course that involves buboes? Yersinia pestis, yeah, and there was another STI that we talked about, Haemophilus ducreyi, that also involved like swollen lymph nodes in the inguinal region and these very painful buboes. So the differential diagnosis doesn't really present so much like chlamydia, even though that's what it is. It doesn't present like gonorrhea. It presents a little bit more like perhaps syphilis, in that you can get some ulceration, and like Haemophilus ducreyi, in that you have those inguinal buboes affecting lymph nodes like that. Late stage is usually more associated with proctocolitis, inflammation correcting the colon, and in this case, you might get some systemic symptoms, fever, malaise, body aches, things like that. So, in terms of diagnosis, there are PCRs for chlamydia. Since we don't do a lot of cell culture, we rely heavily on PCR. Serotyping is usually only for epidemiological purposes. We don't commonly do it, just treat the symptoms. And then just trying to interdifferential exclude some of these other causes of genital ulcers. But these, if these wounds do separate, they are still called chancres, and they present very much like syphilitic chancres. Okay, in terms of diagnostics, your specimen types depend on whatever the presentation is. So if you have like a patient with trachoma, we're going to collect a swab from the conjunctiva, conjunctival swab. If you have a patient with an STI, urethral swabs, or another source. Since PCR is the test of choice for chlamydia, the nice thing about testing for chlamydia using PCR is you can get a urine specimen from the patient. You can catch first void urine specimen, 10 to 30 mils, and PCR is sensitive enough to detect chlamydia from those specimens. So the reason why it's nice to get that first void is, any guesses why?
Is it because it's growing and when you pee, you flush it out?
Yeah, well, what's shed in urine along with the urine itself? If it's first void.
When you say first void, it's like first of the day. First of the day.
Yeah, but also not like midstream either.
All the stuff that settled at the bottom, is that?
Epithelial cells.
Epithelial.
Yeah, because remember like the flow of urine, the high flow of urine is supposed to flush out stuff. That's one of the reasons, like, it's like one of our basic defenses. So when you have a first void of urine, it'll flush out some epithelial cells. And what did we say about this bacteria? It's in terms of like being intercellular, exactly. So if we get like a nice first void urine from this patient, you have a wonderfully non-invasive way of testing for chlamydia trachomatis. You can do microscopy, you can do some culture. The only reasons why you think of to do that these days are if you have some sort of concern for antibiotic resistance, or it's a research lab. But like I said, like if you do culture, you have to have monolayers of cells, probably HeLa cells or some other cell line, and you can look for this really characteristic inclusion body that forms in those cells. So that's a feature that would be diagnostic if you're doing the cell culture. You can also do fluorescent antibody stains. It looks like there's a rapid test available too. I have not heard about this much, but PCR is definitely like the primary way that we diagnose chlamydia. And we get a lot of chlamydia test requests in labs, especially from military personnel. Okay, so moving on to that second organism, chlamydia pneumoniae, done with trachomatis and all the things that can be caused by trachomatis. This is a relatively easy pathogen to study because you don't get a lot of extrapulmonary infections. So with chlamydia pneumoniae, it causes primarily a pneumonia, a lot of cases of pneumonia, and not a whole lot else. Community-acquired respiratory tract infection, human to human, close personal contact. It's not going to survive outside the environment for too long. so usually just transmitted in crowded conditions. This is one of the reasons, one of many reasons why they tell you to wash your hands though frequently, because while it is surviving, you know, you can touch something and as long as someone else comes back and touches that in a relatively short window of time, you can transmit it that way as well. One characteristic you want to take note of is this extremely long incubation time, 3 to 4 weeks. So this is not a rapid incubation window at all, it takes some time. There is no seasonality observed with chlamydia pneumoniae, except to the extent possibly that you get more crowding in cold places in the wintertime, indoor crowding perhaps. In terms of the epidemiology, the population that this is associated with is also kind of remarkable, because we're not talking about babies, we're not talking about the elderly, but we're talking about school-age kids, young adults. So that's kind of notable, something you want to keep in mind. Unfortunately, reinfection is common because you don't get lifelong immunity to this. You don't have vaccines. We don't have super good data on how many cases that we get annually because of the fact that most people don't come in for diagnosis. It's fairly mild. Clinical presentation, pharyngitis is possible as well, but usually we are talking about atypical pneumonia. So what do we mean by atypical pneumonia? Anybody ever heard of walking pneumonia? What's walking pneumonia? Yeah.
It's not so much fluid or blood,
Exactly. And that has implications for its transmission as well. So it's particularly with like college age kids or school kids, or like you guys, students in general, if you get hit by influenza, you're not gonna have a choice. You get that description like, I feel like I was hit by a bus, I feel like I was hit by sunlight, you're out, right? And you cannot go to school or class, so you stay home. But if you have something like walking pneumonia, you feel bad, but not badly enough to stay at home. And because of that, you tough it out, you go to work, and then you transmit whatever you have to other people. So chlamydia pneumoniae and the next pathogen we're gonna look at, mycoplasma pneumoniae, are both like big players in that as well. Chest x-ray is oftentimes pretty distinctive for these atypical walking pneumonias. They look like they're pretty bad, but the presentation is not that severe. In terms of, we haven't talked about radiology yet, and I'm certainly not the person to do it, but how these chest radiographs are interpreted is distinctive. You have this really diffuse, patchy information or patchy appearance due to all this information. Compact with clear edges, and this is not what would be typical for classic or typical pneumonia. We've talked about pneumonia, like other bacterial pneumonias. Do you guys remember any of those other bacteria that are associated with typical pneumonia? Strep pneumo is a big one, yeah. So strep pneumo would have a different chest x-ray and probably a more severe presentation as well. Bacterial pneumonias. In terms of diagnostics for respiratory pathogens, we have a lot of PCR panels that include not just one target, but sometimes up to 15 to 18 different respiratory pathogens. So chlamydia pneumonia would be one of those targets on those respiratory pathogens. It's a very common way of identifying and diagnosing this in the lab. Any questions on, yes sir?
So would the clinical course of this be pretty much self-limiting? Is that why, because it says commonly underreported or underdiagnosed, or is this just empirical pneumonia treatment and then it just manages to take out the chlamydia?
Yeah, I think that's probably a pretty accurate statement. I mean, it's treatable. Chlamydia pneumonia is susceptible to a bunch of antibiotics, but as long as you are functional, people may not go in and trouble themselves for a diagnosis.
And then will it be cleared by the immune system in like a common person,
Yep.
or is it just going to be I've just been feeling shitty for years?
No. This is, yeah, I mean, this will usually go away. I don't know, it's not associated with chronic pneumonia. It might last a long time. So chlamydia pneumoniae and mycoplasma pneumoniae are associated with like longer term coughs. even after you clear the infection, you could be coughing for a while, but not chronic in terms of like, I've had this for a year type of chronicity, just long coughing periods. When I was a kid, I remember my mom had a cough that I swear it lasted like months and months and months, and I was always wondering what was making her sick. But it was a dry cough, and it was very much like this. So I wonder if she had either chlamydia pneumoniae or mycoplasma pneumonia infections. Okay, yeah, let's cover mycoplasma, and then we'll take a quick break. You know what, I lied, let's not, sorry. Sorry, I'm awful. But there's another slide on here too, so let's take a break and come back at 1:53.
In a city like San Antonio where there's a lot of meat, you just ride the party bus, taxis everywhere. But you can do that just about yeah. Yeah, I'm not crazy. It's very it's like, I'm like, yeah, I'm like, looking for a good time. Trust me, you can have a good time. It's just like, crowds, yeah. I wish it smelled like that. We went down the street, granted, I know like this is a thing, but there was guys like shooting up in the alley, house there, and like there was this. It's like somebody dumped this big ass dumpster of trash, and it like for like 2 blocks. They're probably saying, it breathes.
No, you don't. I just keep walking. They got a pop.
Well, I don't know.
They got nothing to lose. They already lost all the team.
Yeah, they ain't got nothing to lose.
Yeah.
Yeah, man. Yeah, that's good.
She hasn't understood.
Oh, okay. Yeah. Well,
Yeah. But like you go there like 7 a.m. They pull you on stage.
yeah. Oh, shit, that sounds fun. I got Germany.
I'm sure every European
Lit, yeah. I don't know. We did a trip up this weekend. We didn't know which one was good. Like, San Antonio was better, and Austin was better. Because I heard Austin was better, but somebody told me Austin sucks. Yeah. I don't know. Yeah, I don't know.
You wanted to do.
Yeah, you know what I mean, yeah.
Talk to your representatives
Exactly.
affordable and this
One of the other ones weren't during the weekend. I was like, how do you get over it?
You're a weekday gal.
Yeah. It's the reason why you're on Thursday night. Okay, don't get it twisted.
That's why I'm going out.
Yeah. Should do that since graduation.
Yeah, no, that's.
Yeah, I just rather wait to get safe. This is the word on the street. I swear. I swear.
I was wondering. I've been sold.
I promise you once, like, they go and they just lose it.
You ain't gotta send me no more. You should be a travel agent. You got so many side gigs after you finish this. You know, you should be a travel agent.
Get a car on this.
Keep that in mind, because I'm looking. I ain't gonna lie.
Just, for real, tell me like the model of it. And look at shit nice and look at the low sticking rows. And if there's like always a time frame, like right before the next one. You're still a year. I love all of it. I'm like 5,000 gas, taking down 5,000 and one time it's like and I'm like, oh, hold on. Like, no. Alright. I'm military too.
Yeah.
Take off that mic. No. I like myself.
That's good. That's a good one.
Yeah, somebody get that
It's not,
That's what my friend did. He called me, this weekend, he was in problems.
Okay, guys. Alright, so about another general atypical bacteria. Just finished talking about chlamydia pneumoniae. Now we're going to talk about this genus called Mycoplasma. It is similar in a lot of ways. We're going to talk about two different species of Mycoplasma, just like we finished talking about chlamydia. One was an STI-related one, and the other was STI/I, as in ocular, and the other caused a lung infection, right? With Mycoplasma, we'll do the same thing. Mycoplasma genitalium is an STI, and then Mycoplasma pneumoniae is also a cause of atypical pneumonia. So first, we'll talk about a couple characteristics of Mycoplasma as a genus. These are among the smallest bacteria. They have been evolving with us for such a long time as intracellular bacteria. Their genome size has been dramatically reduced, so they rely on the host cell to a huge extent. I don't know what their genome size is, but they rely on host cells for ATP, for protein synthesis. I'm sorry, they do have their own proteins, but lipids and some other macromolecules. Very small genome size. An unusual thing about them is that they are not intracellular. The ones we've talked about thus far are intracellular, but one of the main virulence factors these guys have are adhesins on the surface that allow them to adhere to target cells. In terms of their shape, they are pleomorphic, which means they don't really have a regular shape, and that's because they're a very unusual exterior that they have. We'll see a picture of this flask shape that this slide is mentioning, out of a closed quotation, in a couple slides. But if we look at the cell itself, they don't have a cell wall, and they don't have peptidoglycan. So they're basically just one lipid bilayer, and that is it. And that bilayer has sterols. So the sterols are responsible for conferring whatever structural integrity it has, but it is not as robust as most bacterial cells. And it doesn't really need to be either, because it's usually attached to human cells. It doesn't survive long outside the human body. So for transmission, we're talking about the respiratory disease caused by Mycoplasma pneumoniae. It's like you sneezing or coughing in close contact with somebody, and that bacteria is surviving because it's in that moist environment of that aerosolized respiratory part, right? And with the STI forms, similarly, it's not going to be spending much time outside the human body. It's going to go from person to person. So that's how it's able to survive. So very susceptible to osmotic stress, very unique characteristics there. And because it has no cell wall and no peptidoglycan layer, what do you think would happen if you tried to just empirically treat someone who was presenting with either the STI or the respiratory form with a beta-lactam or vancomycin or carbapenem? Why wouldn't that work? Yep, you would be treating somebody there would be no effect on this bacteria. So that's one of the many reasons why it's good to kind of diagnose and get laboratory identification when you can, so you can actually prescribe the right antibiotics, because it would be susceptible to things like aminoglycosides and tetracyclines. As I said, adhesion is critical for it to colonize and establish infection. It is able to vary its proteins on the outside of the cell. Because of that, it makes it difficult for your immune system to clear it, and that has implications for the length of time it takes to clear these infections. This is another one of those slow burn infections that can last a while. Some of these species do produce some toxins, and a small number of enzymes, some of those invasive enzymes we've talked about, like hyaluronidases, but not in huge amounts. And then in terms of culture and identification, this too is target on those big respiratory PCR panels. It's not on this slide, but it's on the next slide. PCR is really important. But you can culture it, it's just gonna take a long time. The time it takes for each bacterial cell to reproduce through binary fission is anywhere up to 16 hours. So 1 to 16 hours depending on the circumstances. Normal bacteria like E. coli has a duration of 30 minutes. So if you wanna culture this, it's gonna take days or weeks in order for you to get these nice big colonies like you can see in this picture there on the right. And when those colonies do grow, they have a super distinctive fried egg appearance. That yellow oak part of that colony is in fact where most of those bacteria are replicating the most quickly, and that's because they can burrow into that agar and produce a small number of hemolysis as well, but that's where they have the easiest time growing. So distinctive appearance when they do finally get going. Okay, in terms of Mycoplasma pneumoniae, this is the #1 cause of walking pneumonia in the US, the atypical walking pneumonia. 2 million cases, 100,000 hospitalizations, it's a significant amount. You can get the pharyngitis, similar to chlamydia pneumoniae, bronchitis, and then we also have the ability for it to cause otitis media, that infection. Talking about the epidemiology a little bit, there are some weird epidemic patterns with Mycoplasma pneumoniae. Every 48 years we get epidemics. I don't know if there's a good reason for that, but it is well observed and something that's probably worth remembering for Mycoplasma pneumoniae. No strict seasonality observed, but it does tend to peak late summer, early fall. Small little increase or uptick in cases around that time period. Similar to when we were talking about chlamydia pneumoniae, you can see like there's a huge number of parallels between these two different organisms. We also have outbreaks in these same populations, healthy, younger populations that tend to crowd together socially. For those of you that are in your 30s and possibly 40s, would you describe yourself as more social now or when you were in your 20s? Massively less, right? So that is a real thing socially. You're getting together a lot more when you're in your 20s. That's one of the reasons why this is associated with folks that are younger. And as you get older, you just stop, you lose interest in hanging out with other human beings, I think. Okay, in terms of clinical significance, very, very similar to chlamydia pneumoniae. Also, very long illnesses, 1 to 3 weeks, sometimes even longer than that. Some of these symptoms are perhaps helpful, but probably not helpful enough to make an correct diagnosis. This minimal sputum production is notable. So you wouldn't be expectorating generally a lot of sputum in these diseases. And the fact that the pneumonia can progress between the lobes and be bilateral just is an indication for it, like hanging out in the lungs long enough and causing and transitioning from a unilateral lobar infection to bilateral lobar. But that doesn't necessarily mean it's more severe. So just like we saw with chlamydia pneumoniae, when those patients have this atypical pneumonia, the chest radiographs might look awful, but the presentation is not that severe. Patients usually aren't that unhappy, and it's not a severe presentation usually. Specimens, we do throat, bronchial lavage, bronchial washings, sputum samples, and just like before, unless you have 5-6 weeks to kill for time, PCR is usually the best way to diagnose it. The second species of mycoplasma is the one that causes STIs, as the name suggests, genitalium. A lot of asymptomatic cases with this as well, and as you'd expect, because of that, it's probably massively underreported. Asymptomatic subclinical infections, but with a prevalence rate of 1 to 4% of the population, that is still a significant public health problem. It's oftentimes described as a silent epidemic. Worldwide, a lot of cases. In terms of its presentation, it presents similarly to chlamydia, non-gonococcal urethritis, NGU, you'll hear that periodically from time to time, NGUs. So So you'll get that, at least in women, mucopurulent discharge, cervix, and men, urethritis. Some other complications that are also pretty much the same complications that you would associate with chlamydia cases. Infertility, cervicitis, salpingitis, which is an infection of the fallopian tubes. And this too, PCR again, so PCR of any of those, like your general specimen types would be appropriate. In terms of the morphology, this is that flask-shaped appearance that I was talking about. You wouldn't see this on any kind of microscopy stain, just would be it. But this is the way they're shaped. This is probably due to the way that they adhere to target cells, this polarity that they have, the reason why they're shaped like that. And then lastly, we have the Rickettsiales. So this is more of a descriptive grouping of these organisms that are in different genera, but they have some common features, so it's kind of nice to study them together. We have 4 different genera: Rickettsia, Aurantia, Ehrlichia, and Anaplasma. We're only going to be talking about Rickettsia and Aurantia, but Ehrlichia and Anaplasma, they're probably good to know as well. We do get cases of ehrlichiosis and anaplasmosis in the United States as well, just not on the hands. So, but if you want to study that in your own time, that would be a super good use of your time, I think. In terms of their morphology, like any of these atypical bacteria that we're talking about, very, very tiny, 0.3 micrometers or so, super small. You can see this is not something you'd normally do clinically, but again, electron micrograph, transmission electron micrograph, you can see that colobacilli shape, and you can see them packed into the cell. So these are back into that category being intracellular, obligate intracellular parasites, also reduced genomes, also dependent on the host cell for many, many, many things, but also still considered true bacteria. LPS has weak endotoxin activity. You've seen that before, that's kind of a common feature. It wouldn't stain very easily, so gibbs stains like Humida stains would also be appropriate. With Rickettsia, this genus, it does have LPS and it does have peptidic lichen, but this other genus, Aurantia, does not. So we won't talk about that too much more, but that has implications for treatment, and that is a defining characteristic of that genus, Aurantia. On your slides, I caught this slight error, so I was trying to enumerate or list out the different diseases associated with those 4 different genera. We've got Rocky Mountain spotted fever, which is the most important one in the United States, arguably, so we'll talk about that in some depth. And then typhus, that's in a different group. So in typhus, these 2 organisms on this chart down here, Rickettsia prowazekii and Rickettsia typhi, are in that typhus group. And it's a little confusing, but Rickettsia prowazekii, that's epidemic or classic typhus, and Rickettsia typhi is endemic or nearing typhus. And then there's also a form of typhus that we don't get in this part of the world at all, called scrub typhus. That's over in East Asia. And that is the only one that we're talking about that's caused by non-Rickettsia bacteria. That's Aurantia, Tsutsugamushi. So, but yeah, so those are the rickettsial and typhus diseases, and then ehrlichiosis and anaplasmosis, which, like as I said, we're not going to talk about more. A common feature of these rickettsial diseases is that they are transmitted by arthropod vectors, and that is a major thing that you need to be aware of in this table. So you've got to be aware of which bacteria are causing the disease, what disease of bacteria cause, which vector transmits those bacteria, and which part of the world they're common in. That's like a minimum amount you need for those 4 different diseases, rickettsial diseases that we're talking about. They have arthropod transmission. They also cause rashes, and they infect endothelial cells. So a lot of the presentation that we're talking about is associated with vascular damage as they're infecting blood vessels. So we'll talk about that. That's a common feature. But if you're just basing your diagnosis on clinical suspicion, you would look at those symptoms, their arthropod bite history, it's a tick bite or some other arthropod, depending on what part of the world you're in, and then the geographical setting. That's why we put so much stress and emphasis on the geographical region as well. For lab diagnosis, we'll talk about this in a little bit more detail for Rocky Mountain spotted fever, but blood, or if you can get it, some biopsy tissue that would have, endothelial cells in them would be appropriate specimens. Serology is useful for diagnosing rickettsial diseases, but keep in mind, as with all antibody-based tests, it's going to take about a week in order for antibodies to start showing up. So, if you want a faster diagnosis, PCR is heavily relied upon. And there are some good PCR panels that have these test targets. And for immunohistochemistry, this too relies on tissue biopsies, which is pretty invasive. So oftentimes these are done only when there's a very serious case or post-mortem or during autopsy. They often take tissue biopsies for testing. And then we'll stain those. Alright, so in terms of these diseases, Rickettsia rickettsii gets its own slide, so we'll talk in more depth about that one. But that's the one that we have here in the United States. It's transmitted by the tick bite, specifically dermacentor. And it does cause a number of cases in the US, so put a dog ear that one. Rickettsia prowazekii is a historically really important rickettsial disease. It's been associated with huge wartime outbreaks transmitted by lice. So anytime you have louse transmission and the inability to access good hygiene and good care, you get some pretty big outbreaks. So it also, try to remember, associate lice with epidemics and outbreaks in resource limited environments where overcrowding is common and it's hard to get good sanitation. This is one of the most severe forms of Rickettsia prowazekii. High mortality rates, up to 60% in estimates, so very high mortality rates. Very important to treat early in order to prevent significant vascular damage and other more serious symptoms. And there's a good book I was going to recommend too, if you guys are interested. Hans Zimmer wrote a book on history of Rickettsia prowazekii. It's called like Lice. Lice is in the title of this book, but it's a super interesting book. It just talks about how the history of mankind has been influenced by this particular disease and outbreaks of it. But worldwide distribution, why does that make sense considering the vectors? Lice are everywhere. Yeah. With Rickettsia typhi, this is a similar version, also causes typhus, but it's a much milder version compared to Rickettsia prowazekii. So very low mortality rates with this endemic murine typhus, and it's also transmitted by a different vector. So we have the flea, and specifically the feces of the flea that get rubbed into an open break in the skin, and then you go on to get this disease that way. Cases do happen in the United States. Almost all of those cases happen in southern Texas. Bear County increasingly used to be more something we'd find out in more remote parts of Texas, but we have been getting cases reported in Bear County, Travis County, where Austin is. So that is one that you might want to also have on your radar. And then finally, Orientia tsugamushi. It spans a huge part of the globe, but not in the Western Hemisphere. So all the way from Siberia over to Southeast Asia, swinging down through the Indian subcontinent. So millions and millions and millions of people are at this risk of infection from scrub typhus, and it's tsugamushi. And this is unusual in that it has an association with mites, transmission through mite bites. Fairly severe presentation. Oftentimes people get an eschar at the site of the mite bite. Damn, it is super rhyming. We've talked about eschars before. What are eschars and what do you associate them with?
Cutaneous anthrax.
Cutaneous anthrax, yeah. So this also can cause a blackened scab-like dried crust with the biting site. And there we have a picture of one such SNR. It's not a great picture, but that is an SNR. That's probably right where at the route of entry. Each one of those 4 diseases that were on the previous slide will have its slightly unique and distinctive presentation features. But in general, the commonalities are that once these bacteria get into the host, they have a tropism for endothelial cells. And that damage is done to, like, direct damage to the endothelial cells because they're intercellular pathogens, so they will cause direct damage. And then once these endothelial cells start dying, you'll get some of these symptoms that you associate with rickettsial diseases. Leaking blood vessels, or permeable blood vessels, leakage of proteins, edema in interstitial spaces, vasculitis, situation of blood vessels. If it gets really bad, you can start getting into some of these symptoms that are associated with death, hypovolemia, disseminated intravascular encephalopathy, shock. So when we do have cases that result in death and fatalities, this is usually how that happens, multiple organ systems. Incubation period is really variable, a few days to a couple weeks after getting the bite. And then in terms of symptoms, there is sort of a classic triad of symptoms that's associated with rickettsial diseases. It's the rash, it's the fever, and it's not really a symptom, but a tick bite itself. So that's like the classic triad. With Rocky Mountain spotted fever, oftentimes, unfortunately, that triad is not present. And if you get a rash at all, that rash might not be there until 2 to 3 weeks afterwards. So for diagnostic purposes, it's not that useful. For that reason, they recommend that if you suspect somebody has a rickettsial disease, you not delay treatment based on just waiting around to see if a rash develops or something like that. You want to treat as early as you can with these diseases. This picture, it's not a great picture, it's a little undersaturated, but you can see this rash, it's all over somebody's torso who has one of these diseases. And I have another picture of Rocky Mountain spotted fever that shows that rash presentation as well. But okay, so Rocky Mountain spotted fever, Rickettsia rickettsii, transmitted by what arthropod? Tick, yes. In the United States, we have, depending on the geographical region, more in the east, we have the American dog tick, germ center, variabilis, I think, and then out closer in the western part of the country, we have a different species, a convector, Rocky Mountain spotted fever. But most of these cases will happen in just a handful of states, these 5 states. We've got Alabama, Arkansas, Missouri, North Carolina, and Tennessee. About 6,200 to 7,000 cases in the US per year. And we have been noticing, the reason why I put this chart in here is because it had nice current data up until 2023, this is on CDC's website. But up until COVID, basically, we've been seeing a pretty steady increase in the number of Rocky Mountain spotted fever cases in the US. And then COVID happened and it clearly shot our surveillance, we had a serious disruption in our surveillance for that. So it'll be interesting to kind of monitor and see what's happening with cases. COVID in general kind of destroyed a lot of our data, surveillance data. Most of these infections have a seasonality to them. That's this bar graph in the upper right hand corner there. Why do you think they have that seasonality from May to, or April to September? Yeah, tick-borne disease, that's where the ticks are out, that's where the people are out. So. And then the American dog tick. So this is a Dermacentor variabilis with its little stripy legs and that white patch on its back. In terms of presentation, it's distinctive from these other rickettsial diseases. Probably the number one thing that's associated with the Rocky Mountain spotted fever, as the name suggests, spotted, is that rash. So most people will develop that rash, but again, that rash might take time. So that's the difficulty with using this as a diagnostic feature. It also has a centripetal spread. What does that mean for rash? Yeah, yeah, moving inwards, centripetal force, so moving inwards. The rash will oftentimes start on the extremities and then move inwards. Kind of the opposite of what you would see with a rash like chickenpox and some other infectious diseases, which would be centrifugal. This is centripetal. Pretty high, this is significant mortality rates if you do not treat, 10 to 25%. And for that reason, CDC has these public health advisories, like educating providers that if you suspect somebody has one of these rickettsial diseases, treat them with doxycycline. There's not a lot of resistance apparently, so doxycycline is kind of like the go-to for treating rickettsial diseases like this. And there you can see like a patient's leg with some really severe rash. It looks like they have some internal bleeding, vascular leakage, petechia formation. It starts looking like a pretty severe disease in these cases. This is a slide that's been in this deck forever. I've been considering taking it out so I don't offend any particular service branch. But I think there is some value in keeping it in here as a mnemonic, since you do have to remember those vectors. So submarines eat rocks, streptitis, neuritis, endophthalmitis, carmisa, and then the vectors themselves may fracture loose teeth. If it's useful to you, use that. I should definitely develop something that doesn't disparage our brains. They need it. They will survive, you're right. Okay, and then the very last genus that we're talking about is Coxiella, and we're only talking about this one organism in that genus, so only Coxiella burnetii. It's kind of an interesting one, and the one that always reminds me of the other disease that we've talked about so far, that we talked about in block 3, that Coxiella reminds me of is Brucella, for lots of reasons. So, but some differences as well. One of the differences, just leading off with, is the fact that this is an intracellular atypical bacteria. It's a tiny little guy, very slow growing. It's more, not that you guys would care as much as providers, but it takes much longer to grow this up than it would for Brucella. It is a Coccobacillus. Gram stain is not something you do this with this either, for the same reasons you wouldn't gram stain any of these other atypical bacteria. The interesting thing about Coxiella burnetii is that it has two structural forms, sort of reminiscent of chlamydia. When we talked about EDs and RBs for chlamydia, which was the form that was associated with the smaller form, was associated with transmission and infectiousness. Yeah, the EDs. Those were the tinier ones, and then the bigger ones are antibodies, metabolically active replicating ones. Those were the larger ones. So same with Coxiella. The small cell variants, the SCVs, are the ones that are infectious. And that's kind of a theme that is good to know. If an organism has two forms, it's usually the smaller one that's the one that's transmissible. The smaller one that is more rugged and environmentally resistant. And the bigger one is the one that is going to be metabolically active and able to replicate. So try to remember that. We see that as a recurring theme with organisms like Coxiella and like chlamydia. In terms of reservoirs, just like with Brucella, we're talking about brucellosis, this also has this is a zoonotic disease, so its reservoirs also in livestock populations. We can catch this disease in similar ways, but there is a slight difference. Because we have this environmentally stable form of Coxiella, the small cell variant, this makes it able to be transmitted in more easily and for longer periods of time. So if you have like a cow that is carrying Coxiella burnetii, that cow, let's say it dies and it leaves a certain amount of Coxiella burnetii, that Coxiella burnetii is environmentally resistant, so it can stay in the environment for a long period of time, and it's also disseminated by the wind too. So we've had outbreaks of Coxiella, we'll talk about acute fever and what it causes, but we've had outbreaks of this disease associated with very long distant carriage of the organism by wind. Actually, I'll talk about it right now. One of the biggest outbreaks we've ever had was in the Netherlands back in 2009 from a goat farm where these goats were infected with Coxsackie burnettii, and the small cell variants were basically being aerosolized and carried by the wind downstream to population centers. And that caused, just like in, there's nothing, this wasn't not biological terrorism or anything like that, it's just normal bacteria just being themselves. And it's sickened about 4,000 people with Q fever in the Netherlands. So you can get outbreaks like that. The reason why it's considered to be a biological threat agent is because of its ability to be aerosolized and disseminated over large areas like that. So it was investigated as a biological weapon by the United States prior to the Biological Weapons Convention, and many other countries as well looked into weaponizing it. Very low LD50, those environmental resistance factors we mentioned. So for those reasons, it was researched for that purpose. So in terms of the epidemiology, this too, oh yes, sir.
Is that LD50 or ID50?
I will confirm that it means LD50. You're probably right though, it probably does mean ID50 since the, it goes down to 10 bacteria. But yeah, I'll confirm that, yeah. Yeah, okay, so in terms of the epidemiology, the disease itself that Coxsackie burnettii causes is Q fever. The Q stands for curry. This was originally characterized in Australia and is still the biggest problem worldwide. It's in Australia where they have most cases of Q fever. Very difficult to diagnose, like brucellosis. You do see cases all over the world, like we talked about with that outbreak in the Netherlands where there are livestock animals. But there are associations with various risk groups that would be the same associations you'd expect for brucellosis, people working with livestock. Transmission is pretty much the same as it is for brucellosis as well. Inhaling those aerosols, being around livestock populations. Another one of these pathogens that can be transmitted through ingesting unpasteurized milk. Can be transmitted by ticks. And notable, when you have somebody with Q fever, this is not considered transmissible person to person. Most of the cases that we have in the United States, you can kind of see, are out in states that you'd associate with livestock ranching. So up here in the Dakotas, Nebraska, Idaho, those places. Okay, in terms of presentation for Q fever, just like brucellosis, very difficult presentation to get a beat on. It's early presentation, general symptoms, non-specific, flu-like high fever, severe headache, chill sweats, fatigue, aches, could be anything, right? And if people progress to one of these other presentations, which depends on the relative entry usually, these can also be mistaken for any number of other pathogens. So just like brucellosis, suspicion of Q fever would rest upon a variety of things, the clinical history, like the endemicity, are you in one of those states that has a big livestock population? Yeah, so the symptoms aren't necessarily going to be that helpful. In terms of vaccines, for going back to brucella, do we have a vaccine for brucella for livestock? We do, yes. We also, there is a vaccine as well for Coxsackie burnettii for livestock. And I think in Australia they have a vaccine for humans, that's how big of a problem it is in Australia, but we do not have a vaccine for Coxiella here in the United States. So we rely on really good clinical picture, and this, it is a target on a biological threat panel. If you've ever worked with the BioFire filmed array panel, the BioThreat one, this is a target on that panel. So PCR is good. There are other PCRs besides the BioFire PCR that has this as a target. We can also use Herod's serum, but same caveat or disclaimer applies, 75 to 15 days after illness. And meanwhile, that person could be getting really, really sick confirmatory test results, they recommend you just treat because the disease could be that serious. This is the last slide that I wanted to cover today. I just wanted to, I know this is super small font, but if you look at the targets on that biothreat panel, you can see we have some bacteria, viruses, and toxin encoding genes. We have covered, for the bacterial side, we've covered all of these at this point, I think. Bacillus anthracis, Brucella militensis, Burkholderia malii, Pseudomallii, Coxiella burnetii, Francisella tularensis, Rickettsia prowazakii, and Yersinia pestis. So we've covered all of those. Okay, we'll pick up on Encephalus actually tomorrow. Any questions from me today? Sorry, can you say that a little bit louder? I think the denominator is people who, like, are, like, this is detected in